The arrangement of membrane proteins. The main basic thing is they're integral and peripheral proteins. The integral ones, they're going in, sometimes even completely through, when they are transmembrane proteins, and when they're the peripheral proteins, they're just on the outside. And some which are spanned through, they are having then an alpha helix, and if it goes many times back and forth, it has then a hydrophilic polar region which is protruding into the fluid, extracellular fluid, the watery part, and the watery part, the cytosol, inside of a cell of a lipid bilayer. And also they are empathetic, so one part of them is hydrophilic, and the other one is hydrophobic. The hydrophobic part is in the center, if they're transmembrane, for example, and protruding in them. And that adds to the asymmetry of the lipid bilayer, that the outside is different from the inside part. Also, the transmembrane proteins also are differently on the inside than on the outside. And all these glycolipids, which are the lipid bilayer, the glycolipids, with their head and their fatty acid tail, which are non-polar in the center and hydrophobic, they are now these glycoproteins also, and they have also a carbon group attached, 2 to 60 monosaccharides, and they add to that whole thing of having the outside a sugary coat of carbohydrates, of these monosaccharides. And that forms a completely a sugary coat, the glycocalyx. And that helps to detect which cells are foreign and have to be destroyed for the human body and which are not. It helps to make them slippery because they are hydrophilic. They at the outside, they're hydrophilic, they are pulling in water. And so it prevents them from drying out in the airways in the gastrointestinal tract and makes them slippery if it are blood cells. And that is according to Gerard Roggan-Senner-Grabowski's Principles of Anatomy and Physiology book on the page 63, but this is no substitute for medical knowledge and also no medical advice. Hi and welcome. I'm trying to learn cytology. Today, arrangement of membrane proteins. And after we've done what we're going to do extensor kappa and strength training, and if you participate in the extensor kappa and strength training, then it's very honest. Membrane proteins are divided into two categories, integral and peripheral, according to whether or not they are embedded in the membrane. Integral proteins extend into or completely through the lipid bilayer among the fatty acid tails and can be removed only by methods that disrupt membrane structures. So the lipid bilayers, these phospholipids standing there, the fatty acid tail, the nonpolar part in the middle, hydrophobic polar part, hydrophobic nonpolar part in the center, and the integral proteins protruding into it or even completely through it. Peripheral proteins, I say the peripheral vision, so that's the vision completely, the furthest center which is able to see, so that's the side. So peripheral proteins, on the other hand, associated with membrane lipids or integral proteins at the inner or outer surface, such as the outer surface of the lipid bilayer, or just the outermost surface which can be seen with the peripheral vision, and can be stripped away from the membrane by methods that do not disrupt membrane integrity. That seems to be important, they say it also by verb part, and can be removed only by methods that disrupt membrane structures to the integral proteins, whatever that means. Okay, peripheral just at the outside of a lipid bilayer. Like membrane lipids, integral membrane proteins are empathetic. So empathetic, one part is hydrophilic, another one hydrophobic. The hydrophilic regions protrude into either the aqueous extracellular fluid or the cytosol, and the hydrophobic regions extend among the fatty acid tails. If it goes through it, the part which is hydrophilic is in water outside and the cytosol inside, and in the center there it is the hydrophobic part, the non-polar hydrophobic part. Integral proteins that stretch across the entire membrane and project on both sides of the membrane are termed transmembrane proteins. Transmembrane proteins are the ones which are going from one side to the other one, so lens through it. Often the portion of protein that spans the non-polar portion of the membrane consists of an alpha helix that is formed by amino acids having mainly non-polar hydrophobic side chains. Okay, the part of the transmembrane protein which is going through the non-polar hydrophobic part of a lipid bilayer in the center is oftentimes an alpha helix, formed by amino acids. Side chains, what do we mean by that? Often, the portion of a protein that spans the nonpolar portion of the membrane consists of an alpha helix that is formed by amino acids having mainly nonpolar, hydrophobic side chains. They say something else, as I say it, as I interpret it. Saying clearly the portion that spans the nonpolar portion. I think they say what I say, just adding there's something to that which wasn't explained somewhere else, so we can't really understand it. In some cases, the protein's amino acid chain loops back and forth across the membrane several times. Hydrophobic alpha helix segments that span the membrane alternate with polar hydrophilic portions that protrude into the aqueous fluid on either side. Yes, that's clear. Although many of the integral proteins can float lateral in the lipid bilayer, each individual protein has a specific orientation with respect to the inside and outside faces of the membrane. The lipid bilayer is not fixed, they said it. They can all wander around within a minute or something. It is not fixed, it's more like balloons, so they can be just pushed to the side. So we can move a bit around there. We can float there. And we are also many of these, but also these integral proteins or transmembrane proteins protruding completely through it have a side which is for the in and outside of a cell, contributing to the asymmetric of the lipid bilayer, but one side is different than the other. Many integral proteins are glucoproteins, proteins with sugary carbohydrate groups attached to the ends that protrude into the extracellular fluid. The carbohydrates are short, straight or branched chains composed of 2 to 60 monosaccharides. So the glucoproteins, these are integral proteins, by having a sugar carbohydrate group attached at the end, which is protruding into the extracellular fluid, consisting out of 2 to 60 monosaccharides. The carbohydrate portions of glycolipids and glucoproteins form an extensive sugary cloud called the glucocalyx. Glucolipids and glucoproteins. The composition of a glucocalyx acts like a molecular signature that enables cells to recognize one another. For example, a white blood cell's ability to detect a foreign glucocalyx is one basis of the immune response. Is one basis of the immune response that helps the body to destroy invading organisms. So the glucolipids and glucoproteins, they have a sugary outside part of carbohydrates, 2 to 60 monosaccharides. And they form in the glucocalyx a sugary part, and on that the cells can identify other cells if they are foreign or part of the body and have to be destroyed or not. In addition, the glucocalyx enables cells to adhere to one another in some tissues, and they can also sometimes adhere from the glucocalyx. And it protects the cell from being digested by enzymes in the extracellular fluid. Connecting to not destroy the immune system, detecting, adhering, protected from being digested, and the hydrophilic properties of the glucocalyx attract the film of fluid to the surface of many cells. It's hydrophilic, attracting water of a fluid to the surface, an action that makes red blood cells slippery as they flow through narrow blood vessels and protects cells that line the airways and the gastrointestinal tract from drying out. Drying out in the airways and gastrointestinal tract and makes the blood cells slippery. Okay, I'm going to think. The arrangement of membrane proteins. The main basic thing is they're integral and peripheral proteins. The integral ones, they're going in, sometimes even completely through, then they are transmembrane proteins, and then there are the peripheral proteins, they're just on the outside. And some which are spanned through, they are having then an alpha helix, and if it goes many times back and forth, it has then a hydrophilic polar region which is protruding into the fluid, extracellular fluid, the watery part, and the watery part is cytosol inside of a cell of a lipid bilayer. And also they are epiphatics, so one part of them is hydrophilic, and then one is hydrophobic. The hydrophobic part is in the center, if they're transmembrane, for example, and protruding in them. And that adds to the asymmetry of the lipid bilayer, that the outside is different from the inside part. Also, the transmembrane proteins also are differently on the inside than on the outside. And all these glycolipids, which are the lipid bilayer, the glycolipids, with the head and then fatty acid tail, which are non-polar in the center and hydrophobic, they are now these glycoproteins also, and they have also a carbon group attached, 2 to 60 monosaccharides, and they add to that whole thing of having the outside a sugary coat of carbohydrates, of these monosaccharides. And that forms a completely a sugary coat, the glycocalyx. And that helps to detect which cells are foreign and have to be destroyed for the human body and which not. It helps to make them slippery because they are hydrophilic. Where at the outside they're hydrophilic, they are pulling in water. And so it prevents them from drying out in the airways. It gets into the cell tract and makes them slippery if there are blood cells. And that is according to Gerard Rogan Senegabowski's Principles of Anatomy and Physiology book on the page 63, but this is no substitute for medical knowledge and also no medical advice. I don't know, I could make it certainly way better if I do it just the second time. But maybe it's also better if I not talk too fast. I'm going to say it second time. And that was the cytology part. If you were here for cytology, you can go to the next part, cytology. We will continue with cytology. If you stay here and participate in the extensor capillary strength training, is that you always have a good and stable standing stance, arms up, the fists, and bring them down. Down and down. We're having 3 rules. The first rule is that we're stopping immediately as soon as there's pain. The second rule is that we go only as far as we can go with a little bit of effort. And the third rule is that we're not overdoing it. We just want to strengthen, not to exhaust. You can use a soccer t-shirt or a towel. Press onto it and bring the hand down. Press equally with all 5 fingers again up. And down. Up. And change. Press. Down. And up. And again down. And we go with the other hand again to the outside. Again, only as far as it goes, without a lot of effort, all pains. And press evenly with all 5 fingers. Outside. And your hand. And out. And weer hand. And down, not from the top. And changing it back. Press and down. And up. Down. And press again. Neutral position. Change. And that goes the extensor capillary strength training. 8 are the phospholipids and the glycolipids. So glycolipids is right here, and the other ones are the glycoproteins. The glycolipids are the one which form the lipid bilayer, and the glycoproteins are the one which are the integral or peripheral proteins. I don't know if I said it wrong, I'm making asterisks. That was now the extensive and strengthening and the cytology part. I thank you very much, and next time.